Hi, my name is Alyssa, and in this video we will be exploring the concepts of classical genetics and looking into how physical traits are inherited from one generation to the next. We'll start by looking at how we determine whether a gene is dominant or recessive for a given trait, and then examine the two terms penetrance and expressivity. We'll finish off by exploring Mendel's laws of genetics, and then look into the experiments that help support the hypothesis that DNA is the genetic material that passes traits down from parent to offspring. After watching this video, you will be familiar with how traits are passed down through generations and how dominant and recessive genes work. You will be able to answer questions that ask you to think about how different phenomena will affect how genes are dispersed throughout the population. We'll start off by looking into patterns of dominance and what genes are and where they are found. Genes are the DNA sequences that determine the physical and biochemical characteristics of all organisms. All genes are found on chromosomes, the genetic material passed from parent to offspring. This image is depicting a karyotype of a human and demonstrates that chromosomes in the nucleus have two copies, one from the father and one from the mother. If we take a closer look at one of the chromosomes from the pair, we can see that there are a number of different genes found on just one chromosome. Sometimes the information from one chromosome is different from the other, and these variations in genes are called alleles. Let's look into this a bit more in detail. Since there are variations in the genes between the two chromosomes, the physical or biochemical traits that the gene defines can vary depending on which two alleles are present. Alleles can be either dominant or recessive. This means that the trait from one allele, the dominant one, can actually overpower the traits from the other, the recessive, and only one trait will be expressed. Therefore, if an individual has a dominant gene, they will express the dominant trait, no matter if there are two dominant alleles or one dominant and one recessive. This means that the only way the recessive trait can be expressed is if both alleles are recessive. This dominant-recessive relationship is known as complete dominance. By having at least two different opportunities for the same gene, there are a number of different combinations for alleles. This is known as the genotype, which describes the combination of genes being dominant-dominant, dominant-recessive, or recessive-recessive. The phenotype is slightly different. This describes how the genes are expressed physically, for example, whether the dominant or recessive gene is expressed. Let's look at how we get different genotypes and phenotypes. This chart is called a Punnett square and helps express visually how genes are combined when the chromosomes of two parents come together. In this case, the genotypes of the two parents have one dominant allele, capital B, and one recessive allele, lowercase b. When these parents produce offspring, there are four different possible outcomes. The first is that the offspring will inherit dominant alleles from both parents. In this case, the genotype is said to be homozygous dominant. Another possibility is that the offspring will inherit recessive alleles from both parents. This is called homozygous recessive. There are two other possibilities, where the offspring inherits a dominant allele from one parent and a recessive allele from the other. Since the dominant allele could come from the mother, and the recessive allele could come from the father, and vice versa, there is twice the probability that this genotype, called a heterozygous genotype, will form. Take note that hetero prefixes mean different and homo prefixes mean same. This should help you remember the difference between these terms. Remember that these genes code for certain characteristics for the offspring. In the offspring having one or two dominant alleles, the color of the flower is purple, just like the parents who both have a dominant allele for purple color. However, the homozygous recessive offspring does not have the allele for purple, and so its color will be white. These tables are excellent ways to visualize not only the different genotypes of offspring, but their phenotypes as well. We've just looked at an example of complete dominance, but there are some cases where codominance or incomplete dominance can occur. What do these terms mean? We'll start by looking at codominance, for example, blood antigens. Codominance occurs when both dominant alleles express themselves into the observable traits, and neither is dominant over the other. In individuals with alleles for both A and B blood antigens, both antigens are found in the blood because these alleles are expressed simultaneously. We can also look at flowers and see that in this flower, pink and white are dominant alleles, and both are expressed to yield a flower with both pink and white sections. Let's now take a look at incomplete dominance. In these cases, neither allele has dominance over the other, and the trait is a mixture of the two. From this Punnett square, we can see that with two dominant or recessive alleles, the flowers are red and white respectively. However, with a heterozygous genotype, the flower is pink, a mixture of both red and white. Now let's examine the terms penetrance and expressivity to understand a bit more about genes that are expressed within the population. Penetrance is the probability that an individual in a population will express a specific phenotype given their genotype. Alleles can therefore be classified based on the amount of individuals expressing that phenotype. If penetrance is full, it means that 100% of the population with that genotype express the characteristics of that gene. Penetrance can also be high or low, and is given a percentage based on whether most or not many of the people in the population have that phenotype. Take the example of dogs with a mutation in gene W that causes cancer. The penetrance of this allele is 75%, which means that 25% of the population having this mutation will not get cancer based on this gene. Now we'll look at expressivity. This describes identical genotypes that produce different phenotypes. If expressivity is constant, then all individuals with that genotype will express the same characteristics, whereas if expressivity is variable, individuals with the same genotype will express different phenotypes. Expressivity is not regarded in terms of population, but is examined based on the individual gene. We can look at the gene for height. We know from experience that people have different heights, but in some cases, even though their heights are different, they can have the same genotype for height. This is an example of variable expressivity. Gregor Mendel is the father of modern genetics and gained most of his knowledge of genes and inheritance from studying pea plants. From these studies, he proposed two laws. Let's look at the first in detail now, the law of segregation, which has four main principles to keep in mind. Mendel stated that genes exist as alleles, which we have already looked at and know as the alternate forms of genes. We also looked at the fact that individuals have two alleles, one inherited from each parent. During meiosis, these alleles separate into individual alleles so that sex cells contain only one allele to be passed down to offspring. These alleles are said to be dominant or recessive, where only the dominant allele is expressed. However, you must keep in mind the exceptions of codominance and incomplete dominance that we covered in this video. The most important thing to remember from this law is that each gamete will only carry one allele, allowing for genetic variation between parents and offspring. Now let's investigate Mendel's second law, the law of independent assortment, which states that the inheritance of one gene is independent of other genes. During meiosis I, recombination of the genes occur. Sister chromatids are combined with homologous chromosomes in prophase I to form tetrads. Here, the chromatids can swap genetic material, called recombination. This results in new and different combinations of alleles found on the chromatid. Mendel first thought that this process was random, and it is when genes are unlinked. However, it is possible that genes can be linked, and therefore their inheritance would depend on other genes, based on their proximity to one another on the chromosome. The important thing to remember about the second law is that inheritance of one gene does not depend on or affect the inheritance of other genes. Finally, we will examine what the genetic material is and how this was discovered. It wasn't until the mid-1900s that DNA was found to be the material that transferred traits from parents to offspring. Initially, it had been thought that this task was accomplished by proteins. In the 1920s, Frederick Griffith performed an experiment in which the term transforming principle was coined and started the road to determining the material involved in this process. Let's look at this experiment in detail. Griffith found that when a strain of bacteria causing pneumonia that was non-virulent and had rough cells was injected into a mouse, it lived. If the mouse was injected with the virulent, smooth cell strain of the bacteria, the mouse would die. If the virulent strain of bacteria was killed by high temperature before injection, the mouse would live, which makes sense. But if the live, non-virulent strain was mixed with the virulent strain that was heat-killed, the mouse surprisingly would die, and live virulent bacteria was found in the mouse. Griffith hypothesized that the non-virulent strain must have somehow gained the ability to cause pneumonia from the heat-killed virulent bacteria. This is the transforming principle. With the information from this experiment, two other groups of scientists completed research that would disprove the old theory that proteins were the genetic material, and support the growing hypothesis that the genetic material was in fact DNA. Now that we have explored the fundamental concepts of genetics, try out some practice questions to test your understanding of the topic. This first question is asking you to think about real-life examples of Mendel's second law. The answer is B. Two siblings may or may not share similar traits for eye color and hair color depending on recombination is a true statement. Remember that during meiosis I, the chromatids can swap genetic information during prophase I. Therefore, the information in one gamete is different from that in the other, and depending on which two combine during fertilization, a number of different traits can be expressed. A is incorrect because fraternal twins are from two different zygotes and are therefore unique. C is incorrect because segregation occurs during meiosis, as stated in Mendel's first law, and D is incorrect because only homologous sites on genes are exchanged during recombination. This second question is asking you to think about how traits can be expressed in the population. The answer is C. If all individuals with the same genotype express the same phenotype, then the expressivity is constant. Great job if you got those questions correct. In this video, we looked at a number of new terms. The genotype is the combination of alleles of a given gene, and the phenotype is how those alleles are expressed in physical or biochemical ways. We looked at codominance, which occurs when there are multiple dominant alleles that can both be expressed at once, and incomplete dominance, where we see intermediate phenotypes because one allele isn't dominant over the other. We defined penetrance as the number of people with a given genotype that express the phenotype, and expressivity is the variation between the phenotypes of the same genotype. Finally, we looked at a Punnett square and saw how alleles combine in a number of different ways. There are dominant alleles and recessive alleles, and their combination can produce either homozygous dominant, homozygous recessive, or heterozygous genotypes. Thank you for watching this video, and good luck studying for your MCAT.